Welcome to part 3 of our series focusing on isotopic measurements using wavelength-scanned cavity ring-down spectroscopy. This webcast introduces isotopic carbon analyses, fast, easy, high-precision measurements of delta 13C from solids and liquids. Brought to you by Picaro, producers of the world's highest performance and easiest to use analyzers. Carbon isotope measurements are critical to our understanding of the carbon cycle, as they allow specific measurements and tracking of fundamental processes such as fossil fuel burning, as well as local and regional effects of temperature over time. Simply stated, carbon dioxide in the atmosphere exists with both carbon-12 and carbon-13 stable isotopes. Carbon-13 is about 1.1% of the total carbon and therefore represents a measurement challenge that fits very well with the performance of wavelength-scanned cavity ring-down spectroscopy systems. Carbon dioxide is photosynthetically fixed by plants, and this process has a general preference for the carbon-12 isotopolog. Plant decay leads to subsequent carbon-12 enrichment of soils and sediments, which over geological time produces fossil fuels. Burning of fossil fuels then leads to carbon-12 enriched CO2 emissions. In plant CO2 fixation, two processes dominate and each discriminates against carbon-13 CO2 differently. The C3 pathway, so called because of a 3-carbon compound intermediate, is considered the older and more abundant. The C4 or Hatch-Slack pathway plants use a 4-carbon intermediate in a process that is considered more energy consuming than the C3 pathway. However, the process is better suited to hotter, drier climates. As global warming and cooling trends occur, the growing regions of C3 and C4 plants can expand or retreat, enabling interesting carbon isotope applications. Overall, the C3 plants discriminate against carbon-13 to a greater extent than C4 plants and have more negative values by about 15 per mil. So, carbon isotopic signature variations are very interesting applications for environmental studies of soils and sediments, and also the organic and inorganic matter in lakes, rivers, and oceans. In addition, the delta carbon-13 values differ among plants, providing a basis for measuring food origin and also adulteration, especially when cheaper C4 contaminants are used in C3 products, corn syrup being one example of a potential adulterant. In order to measure delta carbon-13 values of solids such as soils, leaves, seeds, and liquids such as fruit juices or waters, Picaro and OI Analytical joined forces to develop a transformational technology, the isoCO2 system. This instrument is unique, capable of combusting solids and liquids, or oxidizing organic carbon in water or liberating inorganic carbon in water using OI Analytical's oxidation technology, and then passing the produced CO2 into a Picaro wavelength-scanned cavity ring-down system. The delta 13C values are then measured along with a total CO2 concentration. This system is transforming isotopic measurements. It is benchtop and easy to install and provides high precision competitive with isotopic ratio mass spec. This system is excellent for bulk stable isotopic analyses. Here we have combusted 2 microliter samples of 3 types of edible oil: soybean, sesame seed, and corn oil. Each sample took about 10 minutes for combustion, data acquisition, and analysis, and for these 3 oils we see highly consistent data with standard deviations of 0.17 per mil to 0.5 per mil. Corn oil derived from a C4 plant has a value of -15.08 per mil, while sesame seed and soybean oil have values in the range -28 to -30 per mil, typical of C3 plants. Note that the soybean oil and corn oil are distinguishable from each other at this precision. In a similar bulk application, we have acquired delta 13C values through the combustion of olive oils, premium brands associated with Spain, Italy, Greece, Turkey, Lebanon, and Australia. The IRMS data for these regions has been published previously, and the Picaro data corresponds excellently with every region. In addition, the standard deviation for the cavity ring-down system data is excellent, highly comparable to the IRMS systems, costing 2 to 3 times as much and requiring significantly more maintenance, care, and attention. For those interested in tracking inorganic and organic carbon, we have run a couple of different applications. First, we collected water from 3 different locations around San Francisco. We chose a Pacific Ocean site at Half Moon Bay, a harbor on the west side of San Francisco Bay, a series of estuaries that contains the water draining from about 40% of California, and finally a brackish lake connected to the bay. We would anticipate that the dissolved inorganic carbon values would be different from these highly varying locations, and indeed we see greater depletion of carbon-13 moving from the ocean to the open water bay harbor to the brackish lake. Further, this method was simple. 10% phosphoric acid was added to each sample, nitrogen was bubbled through the water-acid mix, and the liberated CO2 trapped in a mylar bag. The contents of the bag were then passed through the Picaro analyzer, where delta carbon-13 and CO2 concentration was measured. Given the ease of the application, the standard deviation of 5 replicate samples is very impressive, ranging from 0.11 per mil through to 0.28 per mil. And finally, we measured dissolved inorganic and dissolved organic carbon from samples collected from 3 different San Francisco Bay Area watersheds. 2 were collected from watersheds on the west side of the bay, while the third represents the main watershed draining into the south end of the bay. Again, this method is straightforward. An excitainer tube was charged with 4 mils of water from each site. For the inorganic carbon analysis, 1 mil of 5% phosphoric acid was added, and the CO2 measured in the Picaro system. For the dissolved organic carbon, 2 mils of 10% sodium persulfate were added, and the solution held at 98 degrees C. Again, the CO2 produced was passed into the Picaro analyzer. As might be expected, the CO2 concentration liberated from both the inorganic and organic carbon was similar for the 2 western edge locations, and much different from the southern edge. Interestingly, the delta carbon-13 values vary very little for the 3 locations. So in summary, we have shown data that confirms that the ws-crds product is capable of transforming isotopic carbon analysis. The precision is as good as, if not better than, IRMS. The capital and operating costs are both significantly lower. The system is true benchtop and can be located in virtually any laboratory. In addition, it's easy to operate and flexible, capable of performing bulk stable isotope analyses of solids and liquids, and analyzing both dissolved inorganic and dissolved organic carbon in one complete instrument. And once again, if you have any questions about these applications or any other potential applications for this isotopic product, please contact Ian Green or Aaron Van Pelt at the telephone numbers or email addresses shown. Thank you.